Scientists and translational teams developing ADCs are constantly searching for the biological evidence needed to guide their decisions. Sadly, the data they need often doesn't exist yet, and generating it through wet lab experiments is slow, expensive, and limited in scale. At Turbine, we understand this challenge, and we've built a new way to overcome it. Introducing the ADC Payload Selector in Turbine's virtual lab, a platform for running in silico experiments at scale before ever stepping into the wet lab. We combine rich biological data sets like protein-protein interaction networks, cell line expression profiles, mutation data, CRISPR knockout screens, and drug response data to build virtual cells. These are computational models that simulate how real tumor cells behave under different perturbations. By simulating drug behavior in these virtual cells, we help uncover meaningful biological responses and generate predictive data at scale. This enables scientists to investigate hypotheses and patterns that would be extremely difficult or resource-intensive to test in the lab. For example, you can simulate payload sensitivity across hundreds of virtual cell lines representing diverse tumor types, ranging from solid cancers like breast, colorectal, or lung cancer to hematological malignancies. You can break down sensitivity by mutation class, such as comparing BRAF mutant versus wild-type backgrounds. The ADC Payload Selector gives you a full view of payload sensitivity landscapes across these different contexts. It can also model payload combinations, showing which pairs produce the strongest synergy and in which biological settings. In the example on screen, we simulated a payload combination across over 400 cell lines and pinpointed the mechanisms that produced the most synergistic responses. You can try out the demo version of the virtual lab for free with a limited set of payloads and combinations. Cell line names are masked, but you can still explore the core features. A subscription unlocks the full power of the platform, allowing you to simulate hundreds of drugs across thousands of virtual cell lines, including drug combinations, and generate millions of data points tailored to your specific research questions. The virtual lab is designed as an end-to-end application, guiding you through each step. 1. Select models. Start by defining the challenge you want to solve. Whether it's choosing a payload for a new ADC or identifying the right combination partner, the platform adjusts to your goal. 2. Define biological space. Select your virtual cell lines. These represent a diverse range of real tumor biology. 3 and 4. Set up perturbations. Define your simulation inputs, such as drug treatment alone or in combination with a CRISPR knockout or a second drug. Finally, run simulations and analyze. Explore how virtual cells respond, compare sensitivity patterns, and identify standout hits. All your runs are stored in the platform's history, so you can revisit past simulations, track progress, or share insights with colleagues. In upcoming videos, we'll walk you through each of these steps. We hope that Turbine's virtual lab empowers your research by enabling you to run millions of simulated experiments so you can generate the insights you need faster, at greater scale, and with far more biological depth than traditional methods allow. To get started, register for demo access at platform.turbine.ai.